Welcome to the Lions University of South Alabama Eye Research Institute located on the campus of USA in Mobile. My name is Farrell Anders. I'm the treasurer of the Institute and former past president. In 1990, Lions Club International Foundation embarked on an initiative called Sight First that would attempt to eradicate blindness worldwide. Millions have had their sight restored as a result of this initiative. In Mobile, Alabama, local Lions met, and in 1990, the Institute was formed. Over the course of the last 20 years, the Institute has done a lot to reach the goals of Sight First. We have raised approximately $750,000. We have distributed over $150,000 to the research scientists here at the Institute. We have purchased approximately $250,000 of equipment for the scientists to use in conjunction with their research. At the present time, we have $500,000 in an endowed fund. We only use the interest from the endowed fund to fund various projects at the Institute. One of the items of equipment that we have purchased over the past 5 years is a microscope at a cost of $100,000. This was acquired through a grant by Lions Club International Foundation with matching funds by our local Lions clubs. And Dr. Joel Andrews is going to demonstrate the usefulness of this microscope.
Hi, I'm Dr. Joel Andrews, and I'm a postdoctoral researcher at the Mitchell Cancer Institute. But before I took that position, I was a graduate student here at the University of South Alabama, where I did extensive work with the microscope that was obtained by the Lions Club. And I would like to say as a personal note that the acquisition of this microscope made a significant difference in my scientific career because it allowed me to completely reshape my project around the tools made available to us by the Lions Club. And I was able to obtain my PhD and move into the field of microscopy as a result. And the microscope that we have here is a Nikon TE2000 inverted fluorescence microscope. And what this microscope allows us to do is capture images of living as well as fixed tissues and cells. And in the case of my research, it has allowed me to look in unprecedented detail at some of the interactions between a parasite, Toxoplasma gondii, and the host that it inhabits. One of the side effects of infection with this parasite is childhood blindness, a major problem in the third world. It's not as big a problem in our country, but in many developing countries throughout the world, children develop blindness as a result of infection by this parasite, often when their mothers are infected while they're pregnant. And if we look at the scope, you can see this large chamber around it. And what this is an environmental chamber that allows us to control the temperature, carbon dioxide, and humidity of the chamber, which allows us to come as close to possible as replicating what we find inside the human body. This then allows our experiments to be in as natural a state as possible. This microscope is completely computer controlled. It's connected to a camera, and this means that we are able to collect time-lapse video, for example, where we are able to start an experiment and continue to run it and observe what happens over 24, 72 hours, up to a week, as long as you could possibly take it. And in fact, it has a perfect focus system that will maintain focus during this entire period and allow us to really see what's going on in the cells that we're studying. Some of the features of this scope are fluorescent microscopy, where we can use specific dyes and labels to see what's going on inside a cell in brilliant color. And because of the digital camera attached to this scope, we're able to immediately take pictures of events as they're happening. And in my own research with the parasite Toxoplasma gondii, I've been able to capture some of these events in unprecedented detail. And the idea is that by seeing how the parasite interacts with the host, we can hopefully find ways to block that interaction and thereby prevent the blindness caused by the infection. And if you look over here to the monitors, I have some images up of the parasite that I studied. And what you can see here, this is an active infection where you have a infected area here where you can see the parasite in the glowing green color. It's labeled here as the tachyzoite, which is the technical term for the single-celled form of this parasite. And in red around the green, you can see the mitochondria, an organelle of the host cell. And my research focused on the interaction of this parasite with the mitochondria of the host cell, because the mitochondria are what give your cell its energy. And what the parasite is able to do is take those mitochondria and steal their energy, if you will, and use that to its own benefit at the expense of the host. And this parasite drastically changes the host cell, as you can see in some images over here on the other monitor. And in these images, the green color stands for a cytoskeletal protein, that is the skeleton of the cell, and the red is the mitochondria. And you can see here where the parasite has invaded that it has extensively wrapped itself in these fibers and drastically changed the architecture of the cell. I mentioned the video capabilities of this microscope, and these capabilities allowed me to obtain what is one of the centerpieces of my research, and that's a short video in which I was able to demonstrate the parasite actively acquiring a host mitochondria. And as you can see in this video here, we have in the middle of the screen a single parasite of Toxoplasma gondii, and surrounding it in more intense red color are the host mitochondria. And what you will see is this parasite come into contact with the mitochondrion and reel it in towards itself. And I think you'll be able to see this as I play the video. So as it begins, the mitochondria comes into contact with the parasite, and over the course of the next 4 minutes, this mitochondrion is slowly reeled into the parasite, which consumes its energy sources. So in conclusion, the contribution of the Lions Club is incredibly significant for research, not only in my own career, for which I am very grateful, but also more importantly in terms of the problems that they are helping to solve. Research is an expensive business, and tools that can do a lot often cost a lot. But when we have the tools that can really allow us to ask the next generation of questions, then we can come that much closer to curing the problems that are the reason we're doing this research in the first place. So I would like to thank the Lions Club for their consideration and for the generous gift of this microscope. Thank you.
The demonstration that you've seen Dr. Andrews do today is just one facet of the research that's being done by the Institute. This work requires constant support, and we need your financial contribution to make this occur. Thank you so much for your interest in the Institute.